Refactoring involves rewriting a natural genetic sequence for various purposes. Often the goal is to transfer processes from one organism to another, but in other cases the goal is more subtle. In this study, Andy and co-workers rewrite the T7 phage genome to have well-defined gene boundaries amenable to BioBrick-style cloning methods. It was the first introduction to the idea of rewriting a genetic circuit to make it more engineerable through refactoring. The T7 phage infects E. coli and is encoded by a 40 kb genome encoding 55 proteins. It is extensively well studied from the perspective of traditional genetics, and a great deal is known about the role of each protein. Many of these proteins are involved in forming the proteinaceous shell of the phage, while others are involved in DNA replication and packaging steps, lysis of the cell, and a great deal of regulation. In previous studies, the authors had done computational models to T7 phage, but were unable to make predictions about the effects of mutations in the phage. It was clear that subtle aspects of T7 were either misunderstood or not yet identified. Part of the difficulty is that phage often contain overlapping open reading frames in which the coding sequence of one protein overlaps the regulation of another. Such scenarios can give complicated behavior. Their proposed solution is to try to construct a phage that behaves like the model rather than making the model fit the phage. The first step for this would be to introduce sites into the phage that allow them to swap in and out regulation. What they come up with is a variant of T7 called T7.1, and the essential difference between the engineered phage and wild type is the introduction of restriction sites at useful spots of the genome and the repair of overlapping genes into normal-style separated operons. Though the engineered phage is less potent than the wild type in lysis assays, the phage is still functional. What this shows is that you can introduce large-scale perturbations to regulatory regions in a genome without breaking it. This is what we might expect based on our understanding of the regulation in this system. The idea of entirely rewriting genomes to make them more engineerable is an ongoing area of research, as most actively exemplified by the Synthetic Yeast 2.0 project.